Hey guys, in this video we are going to go over replication in eukaryotes. Last video was prokaryotes, now we're doing eukaryotes, and then we're going to talk about telomeres, and we'll talk about DNA repair, the last section of the chapter as well. So when we compare prokaryotes to eukaryotes, in general, it's still the same basic steps. So let's go back to those basic steps. There was initiation, there was elongation, there is termination. This is happening at the origin, and then we're going to use all of these different enzymes that we discussed to undergo DNA replication. So in general, prokaryotes and eukaryotes DNA generation, DNA replication is the same, but there are a few details that differ between prokaryotes and eukaryotes. We talked about that one of those was just the location of where DNA is, right? DNA in the prokaryotes is in the nucleoid, while DNA in the eukaryotes is inside of the membrane-bound nucleus. So DNA replication is happening in different areas of a cell. How many origins are there? We said DNA in prokaryotes is a giant circle, and that giant circle is only going to have one origin. The beginning of where replication starts is called the origin. It's only going to have one on prokaryotes because it's this big giant circle. On eukaryotes, remember we said we had linear chromosomes. We have linear chromosomes, and there might be multiple spots on those chromosomes where we are undergoing a replication bubble and we are copying the DNA. So we have multiple spots of multiple origins of replication for eukaryotes, us, and prokaryotes, bacteria, archaea, only have the one for their big single circle. Speed, this is another thing that I want y'all to know the difference of. We are only about 100 nucleotides per second when we're trying to copy our DNA under the S phase of interphase. Prokaryotes, on the other side, we said that they can copy themselves really quickly. Like they can copy their entire cell in like 40 minutes, I think is how fast an E. coli can create another E. coli. So they can lay down new DNA really, really quickly, 10 times as fast. So they can lay down 1,000 nucleotides per second versus our 100 nucleotides per second. And that's being generous. We may even be even slower than that. Prokaryotes are fast. So their rate is faster, and they only have one origin of replication. I'm not going to ask you about all the different polymerases or the telomerase. They are both going to need RNA primers. They are both going to elongate the strands with DNA pole. It's just slightly different, but big differences for test purposes. One origin here and lots of origins for prokaryotes. Super fast replication for prokaryotes, comparatively slower replication for eukaryotes. So those are the big takeaways I want y'all to know. This video here goes into very detailed takeaways comparing prokaryotes to eukaryotes. DNA replication, he goes into lots. I need y'all to know origins differ and the speed differs. Telomeres. Okay, so we said that we are going to lay down DNA pole 5' to 3'. It's going to attach on to that hydroxide OH negative that's hanging on there, and then it's going to continue to go. Every time DNA replicates, it loses a little piece of itself when it does. And so at the end of the chromosome, we are going to lay down these genes that are non-coding. So we might call them junk DNA. They are non-coding DNA sequences, and they repeat hundreds of times, maybe even 1,000 times, so that every time this thing copies, we lose a chunk, but we're not going to dig into the level of the genes yet until many, many versions of replication later. So we're going to lose a piece of them. There's an enzyme Elizabeth Blackburn discovered, and it is called telomerase, and we use this. So that is on this one, that it's present here for eukaryotes. We use telomerase, and what it does is it tacks on those missing junk segments. So every time we replicate, we lose a piece, but telomerase, the enzyme, will tack on that junk segment and then we can continue to copy the cell. So certain cells will have and use telomerase. Stem cells, those germ cells that produce more daughter cells, like the egg and sperm cells that keep producing more sperm. Those sperm stem cells are operating throughout your adult life. If you're an adult male, your sperm are continuing to make more sperm throughout your whole life. So those particular cells are going to use telomerase because they're constantly copying themselves to make more cells. Other normal body cells, adult versions of the cells, we're not going to have those. So eventually those cells do age out and we will lose chunks of the DNA. They will fall off and eventually they will not be able to replicate anymore. So the stem cells use telomerase, cancer can use telomerase, and that's another way that that can keep copying itself over and over and over again without end. So Elizabeth Blackburn is the one who discovered the function of telomerase and what it does. Moving on to the next section. The last section of this chapter is repairing the DNA. DNA has to check itself before it wrecks itself. So it proofreads. DNA poll proofreads. It said that I think for every billion nucleotides, something like that, that one of them has a mistake. So it does make mistakes. And so DNA poll is going to double check and make sure that what it lays down is correct and we don't have any mistakes. It's going to proofread and make sure, because here, again, C's and T's, they go together. A's and T's, they go together. A's and G's, no, that shouldn't be there. So A's and T's go together. So we got to remove that guanine and put in a thymine in its place. Sometimes we end up with those mismatched pairs. And there are enzymes that can come along and fix them, recognize that that's not what it's supposed to be, and fix them. How do we end up with mixed match pairs? Sometimes it's laying down, it's spontaneous. Sometimes it's a mutagen that has caused this. There are different types of mutagens. There are chemical mutagens. There's also radiation. Some chemical mutagens will cause this mixed matched pair. It will swap out and alter the nitrogenous bases to where you end up with, instead of an A and a T, you end up with a C and a G instead. And that's not what it should have been. It should have been A's and T's in the first place. So mismatched pairs, we have G and a T, and that's not right. That needs to be fixed. So now we're going to fix it, and we replace the T and put in the C like we are supposed to. So we'll proofread, DNA poll. There are enzymes out there that will fix this. There are different other mutations called dimers. It's usually thymine, and that's when thymine bonds to itself. So we said A's and T's go together, right? Well, if you are exposed to things that can cause mutations, mutagens, radiation, whether that is X-rays, gamma rays, or even UV, non-ionizing radiation, it can cause damage to your DNA. Anybody who's been out in the sun for a long time is possibly susceptible to having damage in their DNA cells, in their DNA, from UV radiation. And it can cause this, thymine dimers. And that's where the thymines bond to themselves instead of hydrogen bonding to its base pair across the way. So thymine dimers happen when you are exposed to UV rays, and there is an ability for us to cut this stuff out. So we would nick this, and we can cut it and replace it, or we can come along and just destroy that so that they will bond together. But we can fix that thymine dimer and get rid of it. If you cannot fix the thymine dimer, like this person has xeroderma pigmentosa, it is a condition where they can't fix the thymine dimers. And so it is a recessive, so we said to get a recessive trait that displays as a phenotype, you have to have it from both sets, both mom and dad. So xeroderma pigmentosa, every time this person is exposed to UV light enough to cause damage in their thymine, then they have thymine dimers, and that's going to leave skin lesions on the skin that is exposed to the sunlight. Our last view is of mutations. Sorry, I have a neighbor nest fell down and it has a baby bird in it, and He's hungry and he needs to be fed. I'm gonna feed him in a second. There are different types of mutations. We said mutations can be caused by chemical mutagens. They can be caused by radiation like X-rays, gamma rays. They can be caused by UV radiation. So there's a movie coming out. It's called The Radium Girls, and there is some backstory because it's based on real life. But there's a link here to tell you a little bit about that and the radiation that they were exposed to with radium and how they literally glowed in the dark. They were so radioactive. It's crazy. It's a crazy story based on real life. So check that out, Radium Girls. Different types of mutations. We're going to break them into two groups. We have a point mutation and we have a frame shift mutation. We'll start with the point mutations first. So point means just one little spot, right? It's just one spot. So with a point mutation, the way these work for central dogma, we take DNA and it's going to be transcribed into the message, which is RNA, and that gets translated into an amino acid, which is the basis for proteins. So here we have a strand, and it's going to code for, here's our strand, DNA, and eventually it's going to code for amino acids at the very end. So based on this strand, let's look and see what we have to start with and what we end up finishing with. So AGC is correct, AGC works. GTA, GTT. Okay, so that's a different nucleotide here. Instead of adenine, we ended up with another thymine. And the way these work, you're going to use the codon table, which we'll talk about in the next chapter. And you can see that based on those codons, you code a specific amino acid. And in this case, we have a different amino acid. We have, I'm sorry, this is not a different amino acid. In this case, it is the silent one because GTA and GTT are fortunate enough that both of those code for the exact same amino acid as valine. So even though there is a mutation and we have thymine instead of adenine, this mutation is called silent because it still codes for the same amino acid. So this is a silent mutation. So if you were going to have a mutation out of everything here, the silent one would be the way to go because you still get the amino acid that you want. Again, this will make more sense in the next chapter when we go over the codon table and why we have different things that can code for the same amino acid. For redundancy, we'll get there. But this silent mutation is a change in the DNA, but it still codes for the same amino acid in the end, so it's silent mutation. Missense mutation. So in a missense mutation, we had AGC, AGC, GTA, GTA, CCC, ACC. So this is what I was talking about. Before, we had PRO, amino acid, and now we have THR. We are coding for a different amino acid. So this is a missense when we code for the wrong amino acid. And remember when we were talking about protein structure and we talked about primary, secondary, tertiary, and quaternary protein structure? And quaternary is the most complicated, and hemoglobin was the one that we were saying an example of because it had 4 chains. And remember we talked about how just one amino acid substitution can make a huge difference? That's what we're talking about here. Missense is when you have one amino acid that is coded for incorrectly and it changes the amino acid and it changes the shape of the whole protein. That's what led to sickle cell, was a change in one amino acid. So just like this, it's a missense where we should be coding for this one, but instead we had a coding for a different one because of that 1 point mutation. Nonsense is going to be on the codon table that we're going to see next time. There are start codon, there's 1, and there are 3 stop codons. When we start creating this message, we're going to tell the message to start. We're going to code for these different amino acids, and then eventually we're going to get to a stopping point where it says we are done with this message. Stop coding, the protein is made. So on a nonsense mutation, we end up accidentally, instead of coding for tyrosine, TAC, we accidentally code for TAG, and that's going to produce a stop codon. It's going to stop it. And when we stop it, it's called nonsense. So instead of going all the way along for this long amino acid with lots of different amino acids in there and this long protein, our protein that should be this long accidentally gets shortened to here because we accidentally put a stop codon in there. And instead of being this long, it's this long, and it's not going to work if it's not the full length of the protein like it needs to be. This last group is called frame shift. We're going to go through all of these one more time in the next chapter. But in frame shift, it's just like it sounds. It's going to be either an insertion where we stuck stuff in the strand of DNA and scooched things out, or it's a deletion where we yanked stuff out of the strand of DNA and then the DNA had to come together. Either way, the whole strand of DNA after that shifts. It's either going to shift for an insertion or it will come closer for a deletion, and that whole thing shifts. So that's a frame shift where we have AGC, AGC. We've got the same ones, that's good. GTA, and then CCC. G, we pulled out the T and the A, and it's missing in this one. So that is a deletion. And now if you look from that point over, we're still coding for valine, that's still the same. But these two are not the same as these two because, it's, again, that whole strand of DNA shifted because we deleted some of them out. And now we're coding for totally different proteins. So we'll go over more of these mutations, these same mutations again, and the chapter coming up when we talk about gene expression and central dogma more. If you have questions about this chapter, let me know. Shoot me an email, stop by office hours on Monday and Wednesday, virtually on Zoom, or hit me up on STEM 411.